So today I'm going to talk about genetic maize for polygenic traits in a heterozygous polyploid kiwifruit population. My name is Gibran Tahir, and I'm based in the molecular digital breeding group at Plant Food Research, New Zealand. I'm going to start with some background. Hope you can see it now.
Yes, we can.
Okay, so we already know about the genome cocktails and diploides, which have very intense biology underneath, but it gives us really nice combination of different sets of or copies of genomes. And geneticists are using now the polyploid tools to investigate different important agriculture crops out there, which have different ploidy levels. Our story is around kiwifruit. At Plant Food Research, we have a nice set of germplasm collection, which have a different range of ploidy levels. And kiwifruit is a dioecious crop, so you have a male and female plant, and then you can expect a lot of heterozygosity there. It happens in wild population as well as commercial cultivars as well, because ploidy itself alone is not responsible for elite traits. So there are approximately all the time we have 10,000 plus crosses, breeding parents that are being investigated for various traits. And within kiwifruit, there are 29 linkage groups. And in the market, what you see from as an output of the breeding program is these three types of, mostly these three types of kiwifruit. This one is recently launched, which is diploid yellow with red coloration in the middle. Mostly we see gold, which is also known as yellow tetraploids, and then the green ones are hexaploid. So within the last 10 years, there was a big challenge posed by a pathogen known as Pseudomonas thuringiae actineria, and actually it killed almost 100% of the elite kiwifruit cultivar Hot 16A, which was made by New Zealand breeders. So this was a big challenge at that time, and in the same time period, the tetraploid population surfaced up, which were more tolerant to PSA, and obviously these males were then became the part of the breeding program to cross with elite female parents, so then you can have much more tolerant progeny. So when I started this work as a postdoc at Plant Food Research, we were aiming to map PSA resistance in diploid as well as tetraploid population. So today I'm going to talk about this tetraploid case. So this was a cross with a susceptible female with a tolerant male. We had 230 F1 individuals initially. They were replicated 10 times and planted at two different sites. One is Tipuki, which is like the heart of our breeding center in New Zealand, and then Kerikeri, which is up in north. So the phenotyping was done across 2 years. For the genotyping, we had a kind of a approach that we will capture the Biallelic SNPs, and we will get these dosages, which are kind of displayed up there with 0, with all the small a's as a reference allele. And as you add the alternate alleles in steps, we get these 5 different classes of dosages in tetraploid. So we had that possible through capture sequencing. This was done at Rapid Genomics. They used the 10,000 baits, and the baits were mostly targeted in the exons. So there were roughly 300 markers on each chromosome, which were spaced between 1 to 1.5 MB, so it was quite dense. And we had quite a good coverage across all the chromosomes. So initially we had roughly 700,000 raw variants, but we also looked at the self-relatedness among the population. So for this purpose, we used KGD software, which is actually made for diploids, but we also use it for tetraploid. And we could see that between the parents, we had the progeny nicely here. But then we had some rogues here, which we didn't expect, but that's the joy of working with outcrossing polyploids. We had some issues with calling as well in some individuals, so we think there were lower call rates here. Similarly, At the same time, we were working with Peter Borg, who just talked about this issue in other crops, and he helped us identifying actually these individuals which had higher recombination rates. So, and they were the same individual that we found here, most of them. Also in the polymapr, we could see those red ones which are not fitting the population structure in the PCA plot. So at the end of the day, we ended up with 182 hybrids with high quality sequence data for variant selection. Then the next step was calculating the dosages. So we used UpDoc normal model. It's kind of a empirical Bayesian approach for calculating the dosages. So here I'm just giving you an example of some good and the bad calls that we had there. So the first two plots show that calling was done for discrete genotypes in our population for a particular marker here. We have nice two classes, and then in this one the population has discrete division among three classes. But then in this last plot we can see that something is unexpected there, and UpDoc highlights this, and then we kind of remove those markers. So these kind of biases are either coming from over dispersion or due to other errors. We can actually remove them, and then finally we had roughly 60,000 usable biallelic markers. So then we import them into polymapr. Obviously, it can give you all the classes that it has the ability to show. So then there are more checks in polymapr, so we employed those, and then it's reduced to 45,000. And the classes were simplified as well, like 0 to 1 can segregate like 3 to 4. So that's a good thing. So with this kind of data set, we moved ahead and made the maps, which actually Peter showed in his presentation too. So roughly 39,000 markers. So it was quite a high dense linkage map for a polyploid crop. This is kind of the description of the types of markers. We have a high number of simplex type multiplex, but then the number drops for other types of markers, which help in the development of the maps for homologs. So once we have that done, we did some checks that Peter just talked about. So I don't have to go much into the IBD. So they were pretty okay for us, but there were some issues in the genotypes on those rogues, which we actually removed. So it was an iterative process. We had to do it again and again to really find those genotypes. So that was clear. This on the right side, you can see some the distribution of the markers across the homologs. So on the left side, P1 female, and the right is the male. And then there is a plot here for showing that higher recombination frequencies, which are represented by a higher lot score, are very much tightly present at the diagonal compared to weak lot score with other markers. So the markers are plotted along their recombination scores. Then we went to check the synteny between the markers and the physical position, because the variant calling was done to the diploid genome. We found the synteny is pretty much good there. It's quite linear for most linkage groups, which tells us that genome organization is pretty much similar in the tetraploids. Not much structural changes except a few linkage groups. So on the right, Peter helped us to find some patterns for pairing, and mostly there were. So this plot shows the chi-square values it's an output of the pairing criteria, and most of them were showing polysomic, but then there were some which had preferential pairing. So anything below the threshold, we think it's a case of preferential pairing, and this is consistent with the cytological findings, which found that there is a pattern there for preferential pairing, making kiwifruit kind of a segmental allopolyploid. So that was good, and then we went for QTL mapping. First, we did for the sex. It is a single, it's a Mendelian trait, so controlled by a single gene. And here under the QTL, we can see this homolog 6 from the male is contributed to the sex phenotype. We had the genotypic information coefficient calculated as well, which was at this position was pretty much good. So the score is close to 1 from both parents. it's like unbiased detection for a QTL. And so we were happy, okay, so this is working. But you can see in one of the parent, it falls in the same place. Is because this homolog is actually contributing for the sex marker that we are after. Then we also mapped other trait such as early flowering, which we had 3 QTLs, so it's also important. And finally, we went for mapping the QTL for PSA tolerance. So we had 4 QTLs identified at 4 different groups. And their GIC profiles were pretty good underneath the QTLs, so telling us that our QTL detection is unbiased, so it's good. So, and then we also found 2 QTLs for tissue-specific responses. Then we also calculated if there's an additive effect of the QTLs, and we found that all of them actually working together and giving a percentage variance of 34. So it's a highly polygenic trait, giving quantitative resistance against the pathogen in the field. So it's a typical case of durable resistance there. We found similar results in diploid as well. So it was not surprising, but conferring the hypothesis that the breeders had already there. So the future step was to go and take this data set to the breeding pipeline. In black and white, you have a QTL, you get the capture-seq data, you take out the polymorphism, develop a marker, screen it on the seedlings, either through SNPs or SSRs, and take out those seedlings which don't have the right allele. So what you select are PSA tolerant seedlings, and then you put them in the orchard for fruit traits because it's a 4 to 5 year process. And PSA related deaths they do not happen very quickly. So they take at least 2 years because it's a biotraffic pathogen. So this process has to happen. And what we were thinking, but it's not a low-hanging fruit in our case because we don't have a single QTL. We have a polygenic trait. There are multiple genes and the different homologs are working in a complex way. Also, we had a lack of power to detect QTL that have small effects. And being an outcrossing species, the genetic markers found to be associated with the trait from one cross, they need to be validated for applicability in other crosses. So we developed some SNP markers for HRM profiles. There's some example of different types of heterozygous classes that we had. From on the left side is one parent, on the right, the top is other parent, and then these are Punnett scare. So similarly, we did in the middle of the QTL for each linkage group. So we had different types of classes there, and then we tried to see how the phenotype looks like. So if you look at individual QTLs, so they had a very less impact, roughly around 10%. And we see that, okay, there is some effect, but not very strong. And probably best is to combine the QTLs. So when we combine the alleles on the x-axis and plot the PSA score, we found that there are groups of genotypes which have higher PSA scores. So there are some allelic combination which we can find. And then there are some which are giving tolerance at the lower end. If we look closely into the data set, we do find that these groups have, like the green is showing tolerance and the reds are showing susceptibility. So these allelic combination helped us to find the tolerant ones. And so we can say, okay, it's kind of a success. But when we do all the 4 QTLs, the allelic combinations just are sky high. So you can expect very high combination of there because there are different alleles there. And so we did find that through this marker screening, we did find 84% of the PSA tolerant plants in the field. We could say that, okay, they are on the threshold. Our markers can really segregate them from the bad ones. But we run into another problem because each class is not represented by an equal number of genotypes. So there's not enough representability of genotype class problem. And so we need high numbers of allelic combination to validate this effect. And practicality is another issue because in breeding pipelines, scoring for markers it can be costly. But for the purpose of testing it, we did it does work to some extent. So polyploid tools are now helping us to study key traits in Kiwifruit polyploid breeding program. So there's a dearth in the literature around marker-assisted selection in outcrossing tetraploid species for polygenic traits. And our work actually indicates that there's a strong limitation in employing marker-assisted selection for the complex trait using locus-specific HRM markers. Perhaps the future is that fast and affordable targeted bait sequencing across nursery seedlings may be more high throughput and robust for the search of trait-linked markers in multiple crosses. But in polyploids, there are other ways emerging to look into such a complexity, and I've just pasted a link to that paper, which are more talking about the approach where they have the RNA-seq and other omics data set to nail down the polygenic traits and the underlying genetic and metabolic mechanisms. Lastly, I want to acknowledge all the key authors. Susan Thompson and John McCallum helped a lot in genotyping the bait section and sorting out the variant calling. Louis Gia, who was a breeder, David Chanier and Sue Garner were my postdoc supervisor, and Cyril Brendel is who I'm working currently with at Plant Food Mount Albert. Egmont for funding my postdoc. Special thanks to Peter Bork. He's a very helpful guy, as you mentioned already, and really helped us for polymapr and polyQTLR. David Gerard from American University, he helped us in the UpDog, in the usage of the UpDog software for dosage calculation, and Brian for helping us in the VCFR package for looking at the allele frequencies. That's pretty much. Thank you.